Okay, voice good? Sweet. Well, hi everyone. Yeah, Matt Youngblood. I'm from Ginkgo Bioworks. I'm also here with my colleague Richard. There in the corner. So just throughout the session, if you have any questions about the system I'm about to present, feel free to chat with us. Maybe just a quick background. Some of you might know Ginkgo Bioworks for sort of what Ginkgo has been doing for the last 10, 15 years, which is primarily as a sort of research solutions organization. So companies, government groups would come to Ginkgo with a particular scientific problem, and Ginkgo would sort of act as almost like a black box, do that research and deliver back a strain or a process. But internally, Ginkgo has developed automation and software to perform all of that work, and we are now offering some of that stuff externally. And so we are the group that is building the anaerobic work cell that will be used for this project. And it's a difficult thing to photograph because it's so large, and this is in kind of a small room. But in general, we have 7 different pods that are all connected to one another. And there are integrated devices in each of these pods. So this is sort of a corner view. This is a top view, and I'm going to go into detail on what devices are located in each of these things. But yeah, suffice to say, they're all anaerobic, they're all interconnected, and each of these pods can be atmospherically isolated so that a single pod could be taken down for maintenance or something like that without disrupting the entire system. Let's see. So I wanted to talk a little bit about the anaerobic chamber options themselves. This is, perhaps more relevant for sort of crew that's operating it, but there is a sort of purge mode, so you can you treat it as a purge box where you're just blowing nitrogen in. And this is typically used to bring it down to about 100 ppm oxygen, and then you often switch it into a recirculate mode. These chambers use a copper catalyst. I know a lot of other anaerobic chambers use the palladium hydrogen system to get rid of oxygen, but this uses a copper catalyst. It doesn't require anything additional in the gas. And then there are 2 options. There's a molecular sieve or an activated charcoal filter that you can run the air through to remove various things. Hydrogen sulfide is the one that we worry the most about. And then there's, of course, a HEPA filter just to keep the environment inside those chambers sterile. Before you open it up, so you don't have a wall of nitrogen hitting you, there's a sort of fresh air purge. So you can, if you need to open it up to do some sort of maintenance, you can blow it out with regular just compressed air, and it'll return it to atmospheric oxygen levels before you open it up. The oxygen sensors are for 0 to 1,000 ppm on this system, and so if it's beyond that, you can't really tell what oxygen level it is. There's also humidity and temperature sensors in this, as well as pressure. The devices themselves are integrated with a thing that we call tiles. So tiles are these chassis that can be used to take a particular device and integrate it with other devices. They have a dedicated robotic arm. There's a rail that is in front of a collection of tiles that can pass samples between them. And then all of the necessary networking and control elements are located within an electrical box below there. So this is an example of one of the pods. This is under construction right now, so it looks a little messy, but I can sort of walk here. So right here we have the incubator on the system. This is a thermocytomat that has, it's hooked up for orbital incubation, a 3 millimeter throw, 1,000 RPM, shaking with temperature control. And so we have this rail here with this puck. And so the robotic arm can place samples onto that rail and move it to other devices. We have a thing called a nucleophector, which can do electroporation of cells, a liquid handler called the Agilent Bravo, And then there in the back is a plate reader on the TCAN Spark. And so in general, this rail, moves back and forth within the pod. And then there's another robotic arm that can move samples into this tunnel there at the end. Okay, that's not going to work. I had some video. Oh, no. Okay, this screen is showing something different. I had a video, but I think I'm going to have some IT issues to share that. But this arm can pick up that plate and pass it through, can open up that gate and pass it through there, where it will then go to a tunnel that goes back and forth. And Scott, I might need some help. I clicked on something and I can no longer control it. Okay, there we go. We're back. This is an image of our control software, the sort of visual interface. So you have different operational steps. Say I want to stamp some liquid, or I want to peel a plate, or I want to spin something down. And you sort of drag and drop what labware you want to get delivered to that particular device. And then when you click on that device, there's a set of parameters that appear on the right. So you can specify how hard you want to spin it, what volume you want to stamp, what temperature you want your plate sealer to be at. Another image, there's airlocks if you need to manually pass things in out. These big door panels on the front that can be removed if you need to do maintenance. And then this is a sort of back view of the system. And so this is there's another long rail in that tunnel that connects all of the different chambers to each other. This is a view inside that that tunnel. And then here at the left, you can see that there's this little puck that would bring a sample right up to that door, and then that robotic arm can pass it into a particular pod. Okay, how are we doing on time? Okay, we're trying to end at 9, is that right? Okay. So I thought I would just talk through, just sort of pod by pod, what devices are located on the system. I'm gonna move left to right. We have this kind of funny numbering scheme because we originally designed this flipped upside down and then like locked in our numbering scheme of 1 through 7. So that's why it's 7 through 1. But in pod 7, we have a decapper. So this supports, I'll go to this next slide, micronic tubes that are 0.3, 0.7, or 1.4 mils and externally threaded. So your glycerol stocks or whatever can come in these 96 arrangements of tubes. There's a storage device for the system that just sort of holds various materials. And then we have a cherry picking device. So this has 8 liquid handling channels. And so you can do like arbitrary well-to-well stamping inside this device. It also has liquid sensing abilities. So if you need to pipette things onto, like, say, agar, it's a good device for that. This next chamber has, I sort of talked through it earlier. This is the incubator, that electroporation device, liquid handler, and a spark. Incubator. The plate reader has a variety of features. It can do absorbance, fluorescence, and luminescence. If you need your samples to be, like if you're doing a really long read, this can actually stay warm. It can almost act as like a little mini incubator and shake. And on the absorbance, you can either do like, one particular wavelength, multiple wavelengths, or scan across an entire range. So we've talked through pods 7 and 6. Now, pod 5 is a little unusual. This is the what we call the input-output pod. And so we're almost treating this single pod as a gigantic airlock. So inside of it, there is this storage device, this carousel, and that door is made to be open. Most of the doors are a little bit hard to open. They take like 5-7 minutes to open to undo all these little latches. But on this one, it can be opened up almost just like a car trunk. So it's a little easier to open up, and it's a smaller volume so that purging it is faster. But the idea is that when you're ready to start your experiment, you would walk up to this, you would fill it with your tubes, your empty plates, your things like that, and then close it, and then let that chamber purge and equilibrate until it's anaerobic. And then you can start your process. And you could, there's 2 of these. So there's one located elsewhere on the system. And so it can move all of the work, all the samples to that other internal storage device. And then as waste is generated throughout the experiment, it can return here for removal. This next pod has an HPLC and a breathable sealer. The HPLC we have sort of split into 2 parts. So the injector and the pumps and the place where you actually deliver samples are inside the chamber. But we know that a lot of work on HPLCs it would be really hard to do with gloves on and that kind of thing. And so the column compartment, the refractive index, and the PID, which is a UV absorbance scanning capability, is located outside. And so we have the HPLC pressure lines just like running through the walls of the chamber. So moving on, this pod 3 has a colony picker. This is made by Singer. The way that they pick colonies is kind of interesting. It has this filament that's almost like a weed whacker or whatever you call those, for like trimming yards. It's this long filament that picks a colony, delivers it to your sample, and then it cuts a bit of that filament off. And so rather than having to like sterilize a metal probe each time, it has this sort of consumable part. There's a couple of nice features. You can do a thing where you go down to pick and then drag it across in a particular direction if you need to really scrape up some cells. So colony picker. And then the other device in this pod is the flow cytometer, which is the thermo-attune. This has 2 lasers, a blue laser and a yellow laser, and various detectors. It also can do forward and side scatter. This doesn't do any sorting. It's just an analyzer. But this particular device has they're pretty good at not getting clogged. That's one nice thing about them. They have a wider bore than most other flow cytometers, and they're able to have that wider bore but still maintain this sort of focus of your stream of cells by doing this thing they call acoustic focusing. So there's like sound waves that sort of like push the cells into a single line. It also is an imaging flow cytometer, so it's actually taking little photos of each cell as they pass by. And so there's the potential for bright field images. And then these last 2 pods I'm going to talk about are tend to be on like the logistics side. So we have a thing to peel plates, we have a centrifuge, and then we have a bulk dispenser. So the airlock is right by that bulk dispenser, and so what we've imagined is that media bottles would get passed through that airlock, and then you could, and we'll have a nitrogen line there to bubble those bottles to purge them, and then you can bulk dispense media into your plates there. Bulk dispenser. And then in this pod, we have another peeler, and that's mostly for redundancy. These are kind of finicky devices, and so by having 2 of them, if one goes down, you can use the other. We have another plate sealer. One of the plate sealers on the system is for breathable seals. One of them is for foil seals. And then we have this device called the Tempest. And the Tempest is pretty interesting. This has up to 12 inputs, And so you have these various inputs here, and it can mix a different volume of each of those inputs into every well of the plate. So you could have this, you can end up with like almost like you could do a DOE. You could have like small mixtures of different things in every single well. So you can, in this image, we have several different colors on the inputs, and you can get all these different colors in different wells throughout it. And so we're excited about this because I think it'll open up a lot of opportunities to try big, diverse combinations of different media conditions inside of a single plate. A plate sealer. So yeah, so that's kind of a whirlwind tour of the system. Yeah, questions? Yeah.
Many days of uptime do you expect?
Sure. Yeah, so the question was, what does uptime look like on a system like this? Yeah, just sort of what data do we have there? So this is our first anaerobic system, we have experience in other robotic systems. We think it'll be pretty similar, but we'll, there may be some differences. But our uptime is, so I'll address that in a few ways. One, pick and place errors are pretty rare with our system because we have, go way back to this tile. Because every device has its own robotic arm, that robotic arm is like connected to the same chassis as the device. We can really dial in the sort of movement of plates. So that gets rid of a lot of the kinds of errors you often have with these integrated systems. That said, a lot of these devices just every now and then error out. And so we have a lot of tools available to us to actually remotely control these things or remotely troubleshoot these things without needing to open up the chamber. So we can power cycle these devices remotely. We have all the access to all the low-level controls for the device. And so we've definitely gone days at a time without erroring, but even when there are small errors, you can often recover from them in minutes. I don't know an exact number for that, but yeah. Mm-hmm.
Oh, there is also the metadata.
Yep.
That can be mined.
Yep.
Plans to
yeah, so our software yes, thank you. It's a question about data. Obviously, there is the experimental data itself, HPLC traces and plate reader data, that kind of thing. But there's also, I'll add a little bit to your question, there's device metadata. So this might be, your example is the electroporator. It might give you some information about kind of amperage, the voltage applied, that kind of thing. And then there's the sort of system parameters that you've put in. And then there's also system data, like at what time did a precise event happen? All of that data is made available through our system. So we sort of stream is the correct term. Stream all of that data out, and we will need to work with PNNL to make sure that the LIMS system is able to consume all of that information. But we've had several cases where, yeah, we've been able to make all that information available to different LIMSs, different companies.
Thanks. This is a lot of iron, so congratulations. This is going to become fully operational, just like the Death Star. But quick question for you. We often in our team run into a request, which is, how can we get stuff from the -80 in a more automated fashion? Especially with a lot of the workflows, you're going to need a lot of cell mass. How are you guys thinking about integrating with potentially automated -80s?
So yeah, so there's no cold there's no cold storage on this particular system, and so I think the best course would be to sort of put your cells in and inoculate them kind of as quickly as possible. For other systems that we've designed, it is pretty easy to integrate -20 incubators. -80 is harder. They tend to be almost room size. They're like quite large, the automated -80s. But it is possible. And so that could potentially be a future expansion on this, but not the original plan.
At the moment, we don't have -80 integrated. So this is part of what we'll hopefully learn from here going into the next big system that we're working on.
I'm Rita from Lawrence Berkeley Lab. Nice talk, thank you. I have 2 questions. First, would you please walk through how do we, if we design an experiment from colony picking to the finished result with HPLC analysis, how do we use those 7 parts? Move plates around is still not clear to me. Second question is, how do you perform your instrument QC? Like, how do we make sure the data is reliable after the experiment is done? Thank you.
Well, this is an operational question that I think is still to be determined on who actually writes the protocols that this thing executes. I think the default plan is for staff at PNNL to write that. So contributing users would sort of communicate the experimental plan, and then staff at PNNL would convert that into the sort of language here that is used to control these experiments. Maybe that would change in the future, and maybe this would be it'd be possible to expose this to the end user. But in general, you don't need to worry about which pod a particular device is in. You just describe the different steps that you want to do, and when you draw these arrows, that you want to move this destination plate from your XPeel to your Bravo liquid handler. Our scheduling software is able to figure out the route that plate needs to take in order to get between those 2 devices, whether that's between pods or within a pod. And so our software sort of takes care of that for you, and we'll just move the plate to the next step.
No, they're automatically moved on those tracks. And so just to double down on what Matt said, we're working on trying to convert protocols into that kind of language. So that's in process.
Yeah, the second part on QC. I think this would work like a regular system would. So some devices, will have yearly preventative maintenance events. You should always include controls within your plates. This is another operational detail. Will the control map be determined by end user, or will be determined by PNNL staff? I don't know. But in addition, you could run, like if you just wanted to run a full plate of controls on the HPLC, that would be possible too. So.
So the system can go up to 42 degrees. The humidity you have in the system is enough to maintain the volume of the wells.
So I don't remember the exact temperature range of the Cytomap, but I think it's wider than that. I think it can go hotter than 42 degrees. Richard, do you happen to know the Cytomap, how hot it can get? Okay. But what I was pointing out was the plate reader. The plate reader just happens to have a small incubator within it as well. But when you're cultivating, the plates will primarily be inside the Cytomat incubator. And it has, I think, a pretty wide range. I think maybe it goes into the 50s. There's also humidity control in there. So there's a sort of trough within it that water gets poured into that creates a humid environment within there that hopefully reduces some of that evaporation.
Have you tested it?
We have not tested it anaerobically, but we have tested it with other regular cultures, and it can maintain growth well.
And the volume? Growth, no, not the volume.
I haven't done a full study.
But even if it's not anaerobic, like in the aerobic systems that you say that you did it, still maintains the volume?
We saw good growth, but I didn't do like a full-time course on what our evaporation rate is. But that's certainly a thing that we could do.
Yeah. That would be key.
I'm just curious, the way, I mean, putting all these systems together, is kind of like more newish things, right? The way I would have done it is just to figure out what are the protocols we want to run, and based on that, actually design the system to make those protocols as efficient as possible, right? So I guess what kind of considerations did you guys put into designing the system? Because it seems the protocols are not quite there. Or are they there or not? I mean, I'm just asking. I don't know.
You're just asking questions. I think I'll take this one, Matt.
Yes.
So the question is, what are the protocols? And we purposely wrote the RFP for this system not to have protocols because we did not want to. We're not a pharmaceutical company. We don't want to be constrained by what we can think of now. We want to tap the full creativity of everybody here and the research community to use those instruments to develop new protocols. And so we knew that we wanted phenotypic readings, so we wanted OD. We wanted some sort of analytical, and that's the HPLC. And we decided on the cytometry because we wanted to look at population level characteristics at single cell level, at least cell shape, and potentially with the imaging individual cells. And so the rest is to be determined because we need to develop the protocols and the parameters for the experiments that we're going to be doing. So we didn't, like I said, we didn't want to be constrained to only having one type of media or a limited temperature. So we asked for the most flexibility given a certain set of key performance parameters. Does that answer your question?
Okay.